Hello students, this is Dr. Faizan Mirza. In this video, I'll be discussing with you the structures of cells that include lysosomes, mitochondria, and the cell surface membrane. And in lysosome, I'll be discussing with you how the lysosomes have various functions in the cell. So let's get started with lysosome. Lysosomes are spherical sacs, and these spherical sacs arise from Golgi vesicles. Every Golgi vesicle will not turn into a lysosome, but every lysosome will be made from Golgi vesicles only. These Golgi vesicles that eventually form lysosome have up to 50 different hydrolytic enzymes, and hydrolytic enzymes mean these enzymes will be able to basically break down and digest all biological molecules and harmful chemicals provided to them. Hydrolytic enzyme means enzymes capable of breaking compounds. The size of lysosome is about zero point 0.1 to zero point 0.5 micrometer diameter. Which enzymes do we see are commonly which should be kept in mind regarding the lysosome? So carbohydrates, protease, lipase, DNase, RNase, peroxidase, catalase, all these enzymes are present inside the lysosomes. You can just figure out and just recall which of the substrate do they act on. Carbohydrates is for carbohydrates, protease for protease enzyme for proteins, lipase for lipids, DNA is for DNA, RNase for RNA, peroxidase and catalase, they both carry out the same reaction, and the reaction is working on hydrogen peroxide and breaking it into water and oxygen. One of the functions of lysosomal enzyme is to break down the compounds which are provided to them. So all these enzymes, they work in acidic medium. So pH of lysosome and the contents within the lysosome, they work best in acidic pH. So the pH of lysosome is actually acidic. Comparing this, the pH of cytoplasm. The cytoplasm, which is the site for a majority of chemical reactions in the cell, is having alkaline and a slightly alkaline pH. Coming back. Functions of lysosome. What functions do lysosomes perform? One of the functions of the lysosomes is in phagocytosis. Phagocytes are the type of white blood cells, and these white blood cells are adapted and they have evolved to kill any kind of pathogen which comes to them. Let's see how the lysosomes work there. So this is a bacteria. There are several bacteria present outside your white blood cell. This line here represents your cell surface membrane. I've already discussed this in detail in one of the past paper solving videos of mine as well. So let's just review this again here. This bacteria gets engulfed and gets wrapped up into the cell surface membrane and gets engulfed into the cell, and it is still wrapped up into the cell membrane, and this forms a temporary vacuole and a vesicle we call it as a phagosome. The bacteria is wrapped inside it. This phagosome moves within the cytoplasm, and this moves along into the cytoplasm to meet the lysosome. The lysosome is carrying hydrolytic enzyme. These both fuse together to form a structure called a phagolysosome. Phagolysosome is a structure that ends up forming when lysosome fuses with phagosome. The enzymes of this lysosome start to act on the bacteria, thereby killing the bacteria. In the phagolysosome, once the bacteria has been killed, the digested bacteria will be there inside this vesicle or this temporary vacuole. Useful material will be taken out into the cytoplasm as assimilates for reuse in cytoplasm, whereas all the debris carrying the dead material of the bacteria will be sent out from the cells by the process of exocytosis, and these debris materials will be released outside the cells. So this is how the lysosome works in your phagocytosis. Apart from this, lysosomes also work in removing old and worn out organelles, organelles that are non-functional and are not good at functioning anymore. So your cell gets rid of them using lysosome. Apart from that, lysosome plays a role in PCD, which is programmed cell death, also called as apoptosis. They act as suicide valves, which means they basically will kill each and every organelle of the cell once the programmed cell death occurs, and eventually it will kill the whole cell. This diagram shows to you lysosomes in various stages of digesting the products provided to them. So an organelle might be provided to them. This is about to be digested. This one here has been digested fully. This is cytoplasm that it took up. All you can see the labels here. So this is one of the functions of the lysosome is to get rid of old and worn out organelles. What else is the lysosome doing? from here. So this map actually shows to you how lysosome actually arrives from Golgi. So this is a protein containing vesicle, a transport vesicle coming from the rough ER, and it fuses with Golgi. Inside Golgi, it undergoes modification to be released as a Golgi body. To release from the Golgi body, is released from the Golgi body as a Golgi vesicle. The Golgi vesicle is carrying hydrolytic enzyme. This is a lysosome. So if the lysosome fuses with the phagosome, it forms a phagolysosome. The enzymes start to act on it. And once these enzymes actually have broken down these bacteria and the products have been taken out into cytoplasm for reuse, the debris will go to the cell surface membrane. It will fuse, and exocytosis process will be used to release all these outside insoluble debris outside the cell. Again, this is the same detail shown to you here, how the vesicles from the ERs, the smooth and rough ER, will basically form Golgi vesicles and lysosomes, and eventually lysosome performing their functions. Let's move on to the next organelle called mitochondria. What is mitochondria doing? Mitochondria, we know, is an organelle. It is used in eukaryotes for carrying out aerobic respiration. Inside the mitochondria, we have double membrane. So mitochondria is an organelle apart from the nucleus having a double membrane. The outer membrane is straight in structure or linear in structure, whereas the inner membrane, which is this structure, it is folded inwards into finger-like projections. The inner membrane is folded and the finger-like projections are called cristae. So this finger-like projections are called cristae, and you need to keep in mind that this structure cristae is not cisternae. Some students have difficulty recalling that cristae and cisternae are different structures. Cristae are present in mitochondria, whereas cisternae are present in rough ER, smooth ER, and Golgi. Coming back. So on the cristae, we have a lot of ATP synthesis attached. ATP synthesis are enzymes that produce ATP. Inside the mitochondria, there is a fluid filled called matrix. Between the two membranes, there is an intermembranous space or intermembrane space. The mitochondria is having its own circular small DNA. It is having all the genes which it needs for its lifespan. It produces its own proteins. To produce these proteins, it uses the DNA present inside the mitochondria, and the 70S ribosomes are also present inside the mitochondrial matrix. So they allow proteins to be produced within the mitochondria. So mitochondria actually will not rely on the nucleus for and the rough ER for protein synthesis. It's having its own DNA, it's having its own ribosome. It will synthesize its own proteins within the mitochondria only. Why do you have these inner membrane folded into finger-like projections? This increases the surface area for more enzyme to attach. The enzyme is called ATP synthase. So if more folds are there, more ATP synthase will be there, and more ATP will be produced within the mitochondria. The last structure that I have to cover in this video is cell surface membrane. Cell surface membrane is not exactly an organelle, but yes, a cell structure. Cell surface membrane is a thin permeable layer, and it's not fully permeable. It's selectively permeable or partially permeable. It's 7 nanometer in diameter. It surrounds the cell, and it controls the exchange of materials within the cell and between the cell and its environment. This is the inside of the cell, the cytoplasm, and this region is called outside the cell. So if you see this layer is the cell surface membrane, it appears to be made up of two layers. This is no two membranes, it's one membrane, but the membrane at 250,000 magnification of electron microscope is what your cell surface membrane looks like. Two layers forming one cell surface membrane. And the width of these two layers of one cell surface membrane is 7 nanometer. What your cell membrane is capable of, it prevents the free entry of materials, it's selectively permeable, osmosis, diffusion, active transport can occur, and your cell membrane is having receptors for signaling molecules, for example, your hormones, your transmitters, as well as drugs. They act on the cell surface membrane to bring about changes within the cell. That's it from my side. I hope this video was helpful for you. Thank you so much.